All right, just wanted to talk about this 2014 study that I was on, where we investigate these extended ubiquitins that are the product of mRNA frameshifts, with the main culprit being UBB+1. So especially in neurodegeneration, the accumulation of UBB+1 is a big problem for the cell. A lot of people would assume that it's just the proteasome that's affected, but what we really pin down here is that UBB+1, when it's overexpressed or accumulates in cells, it's causing massive problems with deubiquitinases. And that was the main finding of this study. We have a lot of great work with mass spectrometry and also in vitro assays, as well as yeast systems. And with that, let's dive into the Notebook LM summary of this study.
All right, today we're conducting a results-oriented technical audit of the Krutauz et al. Paper. Our goal is to really reevaluate the core molecular mechanism of UBB+1 toxicity. This is a pathological protein you see in diseases like Alzheimer's. For years, the field has been running on a very specific assumption about how this thing works. Our audit today is designed to put that assumption to the test directly against the data.
Okay, so here's how we're going to structure this audit. First, we'll revisit the established hypothesis, what everyone thought was happening. Then, we'll break down the experimental model they used to test it. From there, we'll critically evaluate the evidence for the old idea, proteasome inhibition, and see if it holds up. Then, we'll look at the data supporting a new mechanism, which is DUB inhibition. And finally, we'll dive into the quantitative mass spectrometer that really cements the new conclusion.
All right, Section 1. Let's start with the central player here, the mutant ubiquitin, UBB+1. We need to be crystal clear on what it is and, more importantly, what the field has long presumed it was doing inside our cells.
So what exactly is UBP+1? Well, it's an aberrant protein that comes from a frameshift mutation in the Ubiquitin-B gene. This results in a ubiquitin protein that has an extra piece tacked onto its C-terminus. For this discussion, we're going to call this whole class of proteins Ubexed. The critical thing is that this stuff accumulates in major neuropathological conditions. Think Alzheimer's and Huntington's disease. That's why understanding its mechanism is so important.
Now, for a long time, the prevailing theory, really the central dogma, has been that UBB+1 is toxic because it gums up the works of the 26S proteasome, which is basically the cell's main garbage disposal. The idea was that it directly blocks it, and that specific claim is exactly what we're here to critically evaluate.
Okay, Section 2, the experimental model. So how did the researchers actually go about testing this? They used a real workhorse model system, Saccharomyces cerevisiae, that's baker's yeast, to dissect the precise function of these extended ubiquitin variants. And here's where their experimental design gets pretty clever. They didn't just look at the pathological UBB+1. They also engineered two other Ubex constructs, one with a 20 amino acid extension and another with 25. This is a really key methodological choice. It allows for a clean, controlled comparison of how the length of that C-terminal extension impacts the ubiquitin proteasome system.
And this brings us to the first key data set, which comes from their supplementary Figure 1. Take a look at this immunoblot. It shows the steady-state cellular levels, and right away you see a critical variable. Even though they're all expressed from identical promoters, the constructs accumulate to very different levels. The UBB+ and the 20 amino acid version are pretty abundant, but the 25 amino acid version is barely detectable. This difference in stability is a crucial piece of the puzzle.
But here's the kicker. Despite those differences in their own stability, expressing any of these Ubex variants leads to the exact same phenotype, a massive pileup of high molecular weight polyubiquitin conjugates. This is a dead giveaway that there's a broad disruption of the UPS. There's a significant bottleneck somewhere in the system.
So on to Section 3. The critical question is that bottleneck at the proteasome itself? We're now going to do a direct audit of that central claim, proteasome inhibition.
You see all those polyubiquitinated substrates accumulating, and it clearly points to a blockage. But is the proteasome really the component being inhibited? The authors decided to test this directly. And here is the first piece of evidence that contradicts the old model. This is a cyclohexamide chase assay, which measures protein degradation rates. Now look at the MyCU BG7625AA variant. That's the one that barely accumulated, remember? It's rapidly turned over. This means it's a proteasome substrate, not an inhibitor. If it were an inhibitor, it would be stable and would accumulate, but instead, it's getting degraded.
And this in vitro degradation assay really provides the definitive counter-evidence.
Here, they used purified components. They took the isolated 20S proteasome core particle and incubated it with the Ubex species. And the result is totally unambiguous. The 20S core actively degrades all of them. There is no inhibition. The proteasome's catalytic core is fully capable of processing these molecules, which directly contradicts the primary claim of the established model.
Okay, Section 4. With direct proteasome inhibition now pretty much ruled out, the investigation pivots to an alternative target, de-ubiquitinating enzymes, or as we call them, DUBs. So the first step is to establish a physical interaction. You have to see if they actually touch. This pull-down assay shows that when they immobilize the UBX variants, including UBB+1, they successfully bind and pull down the proteasome-associated DUB, UBP6. This demonstrates a direct and tight interaction.
And for the functional consequence of that binding, on the left you can see UBP6 efficiently doing its job, cleaving a K48-linked diubiquitin substrate. But on the right, in the presence of UBB+1, that activity is potently inhibited. The mechanism here is structural. UBB+1 has a valine at position 76 instead of the cleavable glycine. This basically turns it into an uncleavable competitive inhibitor that just sits in the DUB's active site and blocks it.
Section 5. So to understand the global consequence of this WB inhibition, we now have to audit the quantitative mass spectrometry data. This is where we get into the dirty details of the cellular ubiquitin landscape.
Right, here we're looking at the absolute quantification data from Supplementary Table 3. In the control cells, total polyubiquitin is around 1,500 femtomoles. But upon UBB+1 expression, that value doubles to about 3,000 femtomoles. This is the quantitative signature of that cellular bottleneck we saw in the immunoblots earlier. And when we dissect this further, we see a significant increase in both K48 link chains, which is your canonical degradation signal, and K63 link chains. This global accumulation of multiple linkage types is far more consistent with broad DUB inhibition than it is with the specific failure of the 26S proteasome.
On to our last section, Section 6. We'll now conclude this audit with a technical summary and discuss the implications of these findings.
The result of all this is really a paradigm shift. The old model of direct proteasome inhibition is replaced. The new model, which is strongly supported by the evidence we've audited, posits that these uncleavable Ubex species are competitive inhibitors of DUBs. So the accumulation of polyubiquitin chains isn't from failed degradation, it's from failed disassembly. It's a crucial distinction.
So, in summary, the technical findings are clear. Ubex variants are substrates of the 20th proteasome, not inhibitors. Their toxicity comes from their function as potent protein-based DUB inhibitors, which causes a global disruption of the ubiquitin landscape. The major implication here is that future research into these neurodegenerative pathologies has to shift its focus. The primary pathological target appears not to be the proteasome, but the de-ubiquitinating enzymes that regulate the entire system. This work effectively redefines a major area of study in the field.